Hi, on this lecture, it's short, it's 3 slides, and I want to talk about dehydration and antidiuretic hormone, or ADH. So, made up this slide, and what I'm trying to show here is this idea of a reflex loop. Sort of a stimulus response loop. Okay, so this is about dehydration. That's what this lecture is about. And antidiuretic hormone and how it works to fix the stress. So if we start, blood and the ECF, we talked before about how the ions, the small things, go back and forth to equilibrium. Now in this case, we're talking about sodium is going to be the major determinant of the ECF volume and the osmolarity. So high osmolarity in the blood, high osmolarity in the ECF. And here's the hypothalamus, and I drew the posterior pituitary on it. There's a little infundibulum from last quarter. So there's the hypothalamus, part of the CNS, and there are osmoreceptors that are associated with it. So there are the osmoreceptors, part of the hypothalamus. That's what's going to be detecting the changes in osmolarity. Well, you might think, how does an osmoreceptor work? Well, first of all, these osmoreceptors are basically neurons. We're in the brain, they're a type of cell, and so these are essentially neurons, specialized neurons. And these neurons can shrink or swell, as we just looked at earlier, depending on solute water differences, shrink or swell. And when those neurons shrink or swell, it opens or closes little stretch gates on their membrane and affects their membrane polarity and their RMP. And they will that will either lead to more action potentials or fewer. So a change in action potentials, I'm getting a little redundant here. Let's say shrinking, could just say change of shape, shrink or swell, changes action potential, AP, frequency. And that then leads to, that information is processed in the hypothalamus. And then you remember that the posterior pituitary is made up of neurons, cell body, and the hypothalamus. The axon extends into the posterior pituitary, and then we release ADH. So ADH is released from the posterior pituitary, an extension of the hypothalamus, depending on whether those osmoreceptors are shrinking or swelling. So if we had dehydration in the osmolarity, that would cause the osmoreceptors to shrink or swell, which would change the action potential frequency, and we release the ADH. Okay. If the osmolarity is high, do you think the cell would be shrinking or swelling? If the osmolarity in the ISF is high, is it going to be gaining water or losing water when it's surrounded by more salt? it's going to lose water and it will shrink. And that will change the action potential frequency. So we release ADH. And ADH then goes into the blood and is carried throughout the body. It leaves the brain, goes through the blood. We know the target of ADH is the collecting duct cells. So ADH comes out of the blood at the kidneys. It's now in the kidneys and it targets cells of the collecting duct. And it causes those cells of the collecting duct to transport water. So remember, we're dehydrated. We want to get the water back into the blood. So water is coming down through the filtrate. I'm drawing water in the filtrate, H2Os. And I want to get that water out of the tubule because it's headed toward the bladder and then the toilet. So we're going to insert aquaporins, and that will allow the water to leave into this steep osmotic gradient. There's more solute out here, clearly. So the water flows toward the higher solutes, 600, 900, 1200, etc. It's out into this medullary osmotic gradient, and then the water is picked up in the vasorecta. It eventually leaves the kidneys through the renal veins and goes back into the blood. And as we add more water, that will do what to osmolarity? Decrease it or increase it? As we add water to it. It will decrease the osmolarity. And that basically completes our reflex loop. We started with high osmolarity and we wound up with lowering it. So we've completed our reflex loop. Back to ADH and the collecting duct. Let's say right here to see the next slide, where we'll look at the details of ADH. So the next slide picks up with ADH arriving at the collecting duct. Oh, no, I added a slide, sorry. Not in my notes. So the next slide talks about vasopressin. So I like the term ADH for antidiuretic hormone. It's an antidiuretic, so it restricts urination. Diuresis is urination, and this is an antidiuretic, so less pee. ADH is the same thing as vasopressin. Just two different names for exactly the same hormone. I prefer the term ADH. One, it's quicker to write, easier to say. That being said, some textbooks use vasopressin, and the illustration that is on the next slide comes from a different textbook, one that I really like, by the way. So just a couple of notes on ADH. Again, it's released from the posterior pituitary when the osmolarity is high. We just went through that. It's the osmolar receptors in the hypothalamus that check the osmolarity. Now, at normal concentrations, this is just sort of a day-to-day regulation of water balance, ADH only regulates water permeability in the collecting duct, as we've seen. So you might think in terms of something like this. If we look at ADH levels on a set of axes, and this being time, maybe it's hourly, and this is the ADH level. So what we're talking about is there's again a high limit and a low limit, and normally ADH is fluctuating in here throughout the day. So when you become dehydrated, you release more ADH. And when you're well hydrated and you don't need any more water, you release less ADH. That's normal. So these are the normal levels. However, and at that low level, ADH has minimal effect on blood vessel constriction. However, if ADH levels increase dramatically, and now we have ADH levels up here, very high ADH. Extend my axes up here a little bit more. Now ADH, in addition to targeting the collecting duct and reclaiming more water, also acts as a vasoconstrictor. And that's where it gets the name vasopressin. Pressin, it's pressing on the blood vessels, it's contracting them, causing them to squeeze and compress the blood to raise blood pressure, but only at high levels. Okay. And what could cause that to go high? Drop in blood pressure. And that, let's, again, this is a review, right? So can you make the connection between a drop in blood pressure and an increase in ADH release? Can you start to connect those pieces in a picture? So you have to stop and think, okay, who is monitoring blood pressure? We've done this early in the quarter. This is part of the challenge. It's a large challenge in this class, is to remember those pieces that we introduced. So this should take you back to the baroreceptor reflex. And the baroreceptors are sending information into the medulla, into the brainstem, which connects with the hypothalamus to the posterior pituitary. Okay, so ADH and vasopressin. Next slide. Okay, so this is a continuation. This slide is showing the details of ADH at the collecting duct cells. And the numbers in the little boxes work pretty well on the left. And let's start with ADH. So here comes ADH. And this figure uses the term vasopressin. But ADH is coming through the blood. It goes into the interstitial fluid. Contacts these cells in the collecting duct. All protein or all hormones out of the pituitary are protein hormones, so they have an extracellular receptor. Okay, that's just shown in the picture. And it shows a second messenger system. We did that either in 160 or 241. We talked about CAMP, and we talked about cyclic AMP and adenosine. Adenine cyclase, which we won't review here, but at any rate, there's a signal pathway that is created. So we can just say that it's a cell signaling pathway from the receptor on the outside of the cell to the inner workings inside the cell. And what happens is that under the influence of ADH in this pathway, there are these storage vesicles. These are membrane-bound little vesicles, and they have aquaporins embedded in the membrane. These are proteins that were translated back on the rough ER to the Golgi, and here they are now sitting inside these cells of the collecting duct. And when ADH comes along, those cells are then exocytosed to the membrane. So the little channels, those little aquaporin channels show up, and then we wind up putting in more of these little aquaporin channels in the membrane. And that allows more water that's in the lumen to be able to enter the cell and we're reclaiming the water. And then that water going to the top part of this cell can then leave. And it leaves the cell out to another aquaporin, out into this high osmotic fluid, very salty, so water readily flows into that. It then flows into the vasorecta, and then that goes back off to the rest of the blood, lowers the osmolarity. So that's the picture that we looked at in the previous slide, but it's showing the details of the aquaporin molecules. Okay. What I like to do when I'm trying to learn something like this is make a little sketch. Make it quick, make it easy, and you're after the concept. So put in perhaps a blood vessel that's delivering the hormone to the cells. Identify the lumen and identify the interstitial fluid so you know which direction is which. Have the hormone arrive at the cell and have that insert new aquaporins on the membrane and reclaim the water. And if you can just sort of sketch that out, it doesn't need to be neat, it just needs to be legible, it needs to be understandable to you. I think that will help you learn this. The next slide, I'll warn you before I put it on, is a busy one. Don't light your hair on fire or anything. And I wish I could enlarge this. On your computer, you should be able to zoom in on this. So, I like it. It ties lots of things together. So let's start. Again, we're not going to panic, but this picture is all about homeostatic compensation for severe dehydration. So I put some suggestions over on the left. When you see a busy figure like this, you have to start breaking it down. You have to pick something, start somewhere, and just kind of work your way through it. So let's start by identifying the stress. Dehydration. Okay. And that should make sense. We have a lower blood volume. We have a lower blood pressure. We've lost fluid. Okay, let's, and then another thing to note on this figure is that it's divided up into 4 columns. So there's this column on the far right, and then there's another column, and then another column. So we have 4 different things going on. And to draw that on a smaller line. Okay, so you're just trying to figure out what this picture is showing. And so you're looking at the shaded columns. ADH is over here on the far right. That's what we looked at. See if this makes sense, if you can read this. And I would enlarge it on a computer. So we have high osmolarity. We had hypothalamic osmoreceptors. Okay, we talked about those. That should make sense. Follow along. And they lead to the hypothalamus. Okay, we've done that. And then the hypothalamus releases vasopressin. Increased vasopressin release from posterior pituitary. Okay, we did that. Targets the distal nephron. Great. And that increases water reabsorption. Okay. And that leads to, follow that arrow over, that leads to increased blood volume, increased blood pressure, voila. Then just pick a couple more lines. So the hypothalamus also, the thirst center is located in the hypothalamus. That becomes active, and that leads to increased water intake. And along with the water reabsorption leads back to increased blood volume. We're just about done with the whole right side. And let's switch over to the left side and see if this makes sense. What are we doing over here? We have a drop, again, back to the stress at the top. We have a drop in blood volume, drop in blood pressure, and we have carotid and aortic baroreceptors. Well, we've talked about those. This should start to look familiar. Cardiovascular control center in the little box, and we have lots of different outputs. We have the parasympathetic, we have the sympathetic. You should be able to work through these boxes and follow along and make sense out of it and get back to increasing blood pressure and blood volume. You have to relax, and I'm not going to ask you to draw something like this, of course, but I do think that it's a skill that you should develop, is to look at a figure and try to decipher it and make sense out of it. You know, so this was our baroreceptor reflex. And what do we have in the middle column? Well, drop in blood pressure. Here are our granular cells. These are the JG cells. And this starts the whole renin cascade. So this whole picture is RAS. So this should look familiar. The granular cells, those JG cells, release renin, angiotensinogen, angiotensin 1, etc., etc. Here's the adrenal cortex. We're focusing on dehydration in this picture. The adrenal cortex releases aldosterone, increased sodium reabsorption, leads to increased water retention, and up goes the blood volume. So we did that fairly early on. So this portion is RAS. And the next slide, or the next lecture rather, we're going to talk about atrial natriuretic peptides. So we'll come back to part of the middle diagram. Okay? So something to note before we leave this is that high osmolarity inhibits release of aldosterone. So where is that shown? Let's follow this through. So back to high osmolarity at the top right, the yellow bubble. So here's high osmolarity. Okay, so follow the dashed line. Comes down, and it comes over here, hangs a left, and goes over and hits the adrenal cortex. So increased osmolarity inhibits. And this textbook uses a symbol of those two little dashed lines to mean blocking or inhibiting. So if you have high osmolarity by itself with dehydration, that will inhibit the release of aldosterone, and you don't get this portion showing up. It's not involved. The rest of it still works, but you just don't release aldosterone. So why not? Why would it be advantageous to inhibit the release of aldosterone if we have high osmolarity? That's the stress. So what does aldosterone do? It promotes sodium reabsorption. If we already have high osmolarity, we don't want to make the situation worse by adding more salt to it. So, okay, that's it for this lecture, and we'll finish off. Actually, I have two more short ones. So the next lecture is just 4 slides, and then 4 slides again. So I'll see you soon. Bye.